Let me see, screen share. Okay. Can you see my screen?
Yes, sir.
Okay. Just as usual, let me take my PowerPoint laser pointer. And okay, maybe we'll start from this point. This is kind of where we stopped last time. Before I go further, anybody have any questions which are nagging them based on what we discussed last time? Before I'll go forward.
Sir, can you zoom out your slides?
Zoom out in the sense, I'm in full screen now.
Yes, sir, but the slides are cutting.
Okay, slides are okay. Let me my sharing again. Maybe something is not right. Just a second. Huh? How about now?
Yes, sir, now it is perfect.
Now it is perfect. Huh? And now if I
Yes,
do
sir.
full screen, how is it now? How is it now?
So again, it is cutting.
Cutting.
So
Okay, okay.
you don't do the full screen. It's fine.
Okay, okay. Just a second. Huh? Let me. Uh, how about now?
Yes, sir. Better.
Okay, okay, okay. So again, let me get my. I need to get my pointer. No. Okay. ठीक है. So, if you remember last time, we started talking about DNA replication, and we said that DNA replication occurs in 3 steps. Okay, we said the first step is what we call as initiation. The second step is elongation, and the third step is termination. Okay, and we looked at some features of initiation, and we looked at how the origin of replications are designed. We said that origin of replications tend to have some repeated sequences, and these repeated sequences tend to be rich in AT, and that allows for DNA to be separated a little easier by the enzyme which actually starts the replication, which is basically this enzyme called as uh, uh, DNA A and DNA B together, which recognize the site and open it up. Okay, when you said that once it is opened, then you have a primer which is added by primase, and DNA polymerase starts to add all the bases in the 5' to 3' direction with proofreading capacity in the 3' to 5' direction. Yeah, we also said that because of this very unique requirement for directionality of DNA polymerase, when DNA replication actually occurs, that at a given fork, you have one strand which is replicated continuously, what you call as a leading strand, while the other strand, which is complementary to the other strand of DNA, is made in a stop and start fashion, in what is called a discontinuous fashion, uh, giving you smaller fragments which are called as Okazaki fragments. This process together is referred to as a semi-discontinuous process. And we also said that to make DNA polymerase's life a little easier, what DNA polymerase actually does is it never actually turns back and synthesizes in the other direction, but it takes the other strand and twists it around so that both the leading strand and lagging strand conceptually are made in the same direction. Okay, which makes things a little more efficient, and this is assisted by this complex which is called as a clamp loading complex, which keeps everything in check. Now if you see this shape here, no, when the DNA has been twisted like this, it looks like a musical instrument. I don't know whether you have seen this musical instrument called as a trombone. Okay, usually it's used very infrequently in Hindi music, but it's used very frequently in jazz music. Okay, and even Latin music, and it has a big little hollow interface which is this shape, and therefore sometimes this model of replication where this DNA polymerase has twisted the strand around like this is jokingly referred to as a trombone model for DNA replication. But essentially, imagine that the replication fork is moving in this direction. This is a leading strand which is made continuously. This is a lagging strand which, even though is synthesized in this direction, is actually made in a discontinuous fashion. Now, if you imagine a circular double-stranded DNA and you start your DNA replication at this point, and your replication forks are moving in either direction, you find that after a while, as replication proceeds, you'll find both the replication forks, if they're moving at about the same pace, will reach a point which is kind of uh, uh, diametrically opposite to where the origin of replication is, or where the replication actually began. And essentially, that is the point at which you'll realize that both these strands here and both these strands here have been actually replicated up to this point. So all we need to do is make sure that the replication doesn't go further from this point, and that the two forks actually meet each other. And that occurs at this junction which is called as a termination sequence. Okay, and at the termination sequence, there are a lot of proteins which bind to this junction, and these proteins are basically referred to as TER A, TER B, TER C, and so on and so forth. Okay, uh, but what you find in this region is that the DNA tends to be rich in GC. Okay, and because the region is rich in GC, what happens is as the replication forks start to come into this area, either in this direction or in this direction, you'll find that the topoisomerase or the gyrase, which is moving ahead of the replication fork and trying to separate the DNA, now finds more and more difficulty in separating DNA because DNA regions have a lot more hydrogen bonds. They are rich in GC. So that what that does is that it kind of slows down the replication fork a little bit, because the gyrase is working a little slowly, or the helicase is working a little slowly. And what that essentially does is that in case one of those two forks is moving slightly faster than the other, this slowing down kind of covers for that, and both the replication forks tend to meet just almost opposite to the origin of replication, because they both kind of slow down here. Okay, it's almost like if you are going to this iMagica or something, no, you do this roller coaster ride. Initially, if you remember the roller coaster ride, the roller coaster is kind of with lot of effort taken up the hill, and then it is left from there, and basically it uses the force of its momentum and gravity to take you down all those loops. And as the ride ends, if you realize, okay, they put these little brakes at the bottom, so that suddenly as you come back to where you started with, the roller coaster becomes really, really slow. Okay, so imagine something like that. These sequences here kind of slows down this roller coaster, which is coming from either side, and make them kind of meet slowly and reach each other, and basically then the two replication forks kind of merge into each other, and the replication is completed. Okay, but uh, and and there's a protein here which is called as a TUS protein, which also helps in this. TUS stands for termination utilization substance, okay, or terminator utilization substance. That protein also assists in this slowing down of the replication forks and the completion of the replication. But you might also imagine for a second that both these strands here are actually double-stranded DNA, which is coiled around each other. And now, as you are doing this replication, what is happening is on each side, you actually end up getting uh, two pieces of double-stranded DNA, right? Here, I have the original template which is being replicated, so that will become a double helix DNA. Then I have the other strand which is replicated in a lagging fashion, which will also become double-stranded DNA. Similarly, in this direction of the replication fork, one strand is read in a continuous fashion, that is replicated, that becomes double-stranded DNA. Then the opposite strand is replicated in stop and start, Okazaki type, and that also over time becomes double-stranded DNA. So now, as you go up to this point, you have to realize there are two sets of double-stranded DNA here and here as everything approaches this point. And when you have helical structures like this, both of which are being replicated, when that fork ends up at one point and the replication forks meet, and then the replication is completed, you'll find that the two sets of double-stranded circular DNA are now entangled. Okay, and they are like locked one inside the other. Okay, almost like those chains that you have, no, where one gets locked into the other, and these type of chromosomes are referred to as catenated chromosomes. So catenated chromosomes are where you have one ring-like structure locked into another ring-like structure. Okay, and this has happened because initially both of them were coiled around each other. Then they both got replicated, and when that happens, there is no way that you will get these two strands separated when everything meets here. They'll both end up giving you two sets of double-stranded DNA which are circular, but we are locked onto each other, and they will not be able to separate. And the only way to separate them is to use an enzyme which is referred to as topoisomerase. Okay, this is also a type of kind of gyrase. If you remember, we talked about gyrases. We said that they cut DNA to relieve the strain. Either they make single-stranded cut or double-stranded cut. Here also, that same enzyme makes a cut in one of those and then lets the other one pass through it and then reseals it. And when that happens, then you end up getting the two circular pieces of DNA which are now separated. Okay, so for circular DNA, as replication ends, you need the activity of this enzyme called topoisomerase in order to kind of separate the two replicated circular pieces of DNA, each of which is double stranded. Okay, everyone clear with this concept of how
So,
things can get? Yeah.
so the topoisomerase or the gyrase which we used earlier
Mm-hmm.
to separate the DNA, so it's the same
Correct.
topoisomerase or is some different strain of it?
No. It's a same variety. Actually, in bacteria, no, there are I think a total of four topoisomerases they have. Okay, uh, topoisomerase 1, 2, 3, and 4, and all of them work slightly differently from each other. Okay, and this is done by topoisomerase 2. Topoisomerase 4, which belongs to the family which cuts both the strands of DNA. Okay, so you'll cut this strand here, allow this DNA to come out, and then reseal this with the original one. Okay, but these are the same enzymes which also can take part in the earlier process of relieving the strain when replication is going on. Okay, so enzymatically they are no different, okay, than the other one. Okay.
Okay, sir.
Now the reason, can anyone tell me the reason they are called as topoisomerases? Anyone want to guess why the name topoisomerase?
Sir, I can only guess the word
Yeah, yeah,
topography,
yeah,
something like.
yeah. See, what happens is when you have double-stranded DNA, no, then different pieces of DNA, okay, which are, let us say, circular, uh, on one hand can be different from each other based on sequences. Okay, so if the sequences of two circular double-stranded DNA is different, we call them basically two different types of DNA. But you can also have a situation where you have two molecules of circular double-stranded DNA whose sequences are totally identical. Okay, but the only difference between the two of them is the number of times they are wound around each other. Okay, uh, either one is relative to the other more supercoiled in a positive fashion, or one is more negatively supercoiled with reference to the other. Now, in this case, if you look at the two pieces of DNA, because their sequences are identical, they are actually not different pieces of DNA in a chemical sense. But because the number of times they are wound around each other is different, then these two pieces of DNA are referred to as topological isomers. Okay, where they differ from each other not in terms of sequence, but just in terms of how many times they are wound around each other. Okay, are they stably wound, are they overwound, or they are underwound? Now, when topoisomerase cuts the DNA and allows one strand to pass over the other, etc., what it is actually doing is making new DNA, which is relative to the old DNA, more supercoiled or more positively supercoiled or more negatively supercoiled. So what it is generating is actually topological isomers of the original DNA. Okay, and that is why the enzyme is referred to as a topoisomerase. So topoisomerases never do anything to the actual sequence of DNA that they are working on. All they do is take the DNA and either overwind it or underwind it, and by doing so, they'll generate a topological isomer. Now look at it in this case. In this case, I'm not even overwinding or underwinding. All I'm doing is cutting this DNA, allowing this DNA to come out and putting this together. But catenates are also considered as one type of isomers of these two pieces of single double-stranded DNA. Okay, remember, they are both circular, right? So in that sense, they both have a sequence, but when they are locked into each other, the sequences are still the same. The only difference is now they are topologically different in that they are locked into each other. So topoisomerase is only unlocking that locking. It is not changing the sequence. In this case, it is not even adding supercoils, but it is going from what is called as catenated forms to isolated forms. And in that regard, that is also a type of topological isomerization. Okay, and that's why these enzymes are referred to as topoisomerase. Is that clear? Everyone, is that clear? What these
Yes,
Yes,
sir.
sir.
enzymes do? So they change not the sequence, but only the form of DNA, or what is called as the topology or topography of DNA. Okay, and then to summarize now, basically, if you recall, we said, okay, the reason these processes work very well is because the DNA polymerase, which is doing the polymerization, even though it works very, very fast and adds 1,000 bases per second, it also has proofreading capacity. And because of that, it is able to correct any wrong insertions that it does. Not only that, after this is done, we have a repair system which does what is called as repair of anything which is not matching and corrects that damage. And because of that, what you find is overall replication proceeds at a rate of making only one error in what is called as 100 million bases. 100 million bases means close to 10 raised to 8 bases. In fact, a lot of people say this is 1000 million bases also, 10 raised to 8 to 10 raised to 9. And that's why this entire process occurs with very high fidelity, or what you call as extreme accuracy. Okay. Now, here is something very interesting, a little puzzle for you. Okay, let us see if anyone can, think about this and let me know. Now, we said that the E. coli genome has, and for just for numbers, 4 million base pairs. Okay, which means in order to complete the replication, I have to actually add 8 million bases, right? Out of which 4 million will base pair with one strand and 4 million will base pair with the other strand. In the end, to give me 2 replicated E. coli genomes, each containing 4 million base pairs. Okay. Now, if we assume that DNA polymerase is adding 2000 nucleotides in each direction. Okay, why did I say 2000 nucleotides in each direction? Remember, it can add 1000 bases per second, but we also know that at any one replication fork, there's one polymerase here and there is one polymerase here. Okay, so each, if it is adding, okay, 1000 bases per second, okay, it probably is not right, because this will add much faster than this one, because it has to stop and start. But just for argument, let us say both are adding at 1000, then in this direction I can probably add 2000 per second. Okay, in the same way, in the other direction also I can add 2000, correct? So the total rate of addition when replication is occurring from the origin of replication in a bidirectional fashion, where each side you have got leading strand and lagging strand, and both are adding at 1000 per second, the total rate of addition will be 4000 bases per second. Okay, which means it is 40,000 bases in 10 seconds, or 240,000 bases per minute. So now if I divide this many additions that I need by this, I end up finding that the time needed for replicating the entire DNA is about 33 minutes. Okay, and this I'm talking about top speed. Okay, top speed where I don't do a lot of turning back, do proofreading and everything like that, assuming everything is going perfectly. Okay, now given that fact, and given the fact that many times E. coli is said to have a doubling time of 20 minutes, how do you think these two match up? If at the top speed it is going to take 33 minutes for the entire DNA to replicate, and I say E. coli doubling time is 20 minutes, I have to also give E. coli time to live, before it tries to replicate. So presuming E. coli spends some time making nucleotides, okay, making all the enzymes which are needed, and after that thing is all there, it is going to take 33 minutes. How the hell can E. coli have a doubling time of 20 minutes? Okay, can anyone want to guess how this works out? That sometimes you actually can get replication time of 20 minutes. Okay, let your thoughts fly now. Okay, ऐसे कैसे हो सकता है? Take your guesses. Okay, all guesses are good. Anyone? Anyone want to guess? Hello? Hello?
Yes, sir.
Yeah, anyone want to guess? Come on, come on. Come on, come on. Some guesses. This is very out of the box thinking now. Okay, you have to do. Okay, how can this potentially be possible? Come on, take guesses. And even if you Google now, you'll not get the answer. Okay, on the side, no? If you're trying to Google and stuff, you may probably not know how to search for it. Okay. So you need to actually think through and take a guess. It doesn't matter, no. Sometimes you have to go out on a limb and and give guesses. It doesn't matter if they're totally off the cuff.
Sir, the 4 million base pairs number, I mean, all these numbers are theoretical, right? I mean, no, we sometimes say, okay.
No, people have taken out E. coli genome, okay, from E. coli and actually sequenced the entire DNA. And that actually is 4.6. It is not even 4.
Yeah.
It is slightly more, okay. I am making your life even easier by giving you a smaller number. Actually, then this time would have been a little longer, okay.
Yes.
It would have been longer by, about maybe another 15% of 33 minutes. Okay, so it might be 35 minutes or so. So guess. Take a guess. How can this happen? Come on. कौन है ये ग्रुप में? शुभम,
Sir,
विनय, हाँ, guess, yeah.
sir, I think like the first replication of it would take 33, and then after that, after like the first replication, when the second cycle starts, it would take a lesser time.
Why will it take a lesser time?
So because already like the nucleotides are present, so the addition would be faster as compared to the previous.
See, all this is based on. I've never accounted for time to make nucleotides here. Okay, here the DNA polymerase adds at 2000 nucleotides per second. Okay, in each direction, and that is the top speed. Okay, and I'm keeping the top speed. Okay, so even if all the nucleotides were there, you cannot go beyond this speed. Okay, and I'm giving you that benefit of the doubt. Okay, even at the first time when replication occurred, it actually occurred at the fastest possible speed. I understand what you are saying. ऐसा रिले के जैसा है. 400 मीटर ऐसे ही भागा मैं. Okay, I can probably run faster than if I run 100 meters stop, then again run 100 meters stop, and that will take, every time I have to get going and all. Okay, but here I've given you the top speed. Okay, so here is a little, what you call out of the box thinking. Okay, what happens in real life, is many times when E. coli is multiplying, and DNA is being replicated, something very interesting happens. As the DNA genome replication begins, and you start from the origin of replication and go in either direction, you will realize that the first thing that has already been replicated, okay, is actually the origin, correct? So if I look at this theta portion here, this is one double stranded DNA, original. Then there is another one which is being replicated. But in both of them, the origin of replication has already been replicated, correct? So what can potentially happen is that as this original circular DNA is being replicated, the new DNA which is formed, that can also get started to replicate, okay, even before this original fellow gets replicated, okay. And in real life, sometimes it is theorized that that actually happens in case of E. coli, okay. So in a sense, what I think it was Shubham who said, okay, next time it will take lesser time. You might argue, okay, this is a way. What you can do is not wait for one DNA to get replicated before you start the other one. So if the original fellow took 33 minutes, I now don't have to start my next round of replication as starting after 33 minutes. My next round of replication could have started 5 minutes into the first replication, which means after 38 minutes, I actually have 4 copies. Are you seeing? When that happens, then sometimes the actual observed, DNA doubling time will work out to be less than what the speed of polymerase tells you. Okay, everyone, is it clear? What I'm trying to tell you?
Yes, sir.
So as the original chromosome gets replicated, you don't wait for the replication to get over before you start another round of replication. That double-stranded DNA is already again starting to get replicated because you have the origin of replication. So many a times in E. coli, this happens simultaneously, and therefore the actual observed doubling time ends up being slightly less than what you calculate simply based on the speed of DNA polymerase. Is that clear, everyone?
So there was a
Yes.
there was an answer in the chat box
Yeah.
from Vidya. She said it may be because there are more replication forks.
No, replicate. See, now if you look at what I just said, no, each circular DNA has only 2 forks of replication. Okay, because in E. coli there's only one origin of replication, so everything can start from there, move in either direction. But when the DNA starts to get replicated, before the first double-stranded DNA is completely replicated, then you might find that as this is appearing, you might actually end up seeing more replication forks than what you should be saying. But that is not because there are more replication forks within the circular DNA. Okay, because E. coli has only one origin of replication. So technically speaking, that answer is not entirely correct. My number of replication forks per circular DNA is still always 2. Okay, but it might appear when a DNA which is not completely replicated is being replicated again, then the structure you might see more replication forks. But for every given circular DNA, there is only one, which moves bidirectional. So you end up getting 2 replication forks moving away from each other every time. It cannot be more than that. Is that clear?
Yes, sir.
Who had answered that? Vindhya.
Yes, sir.
Who? So Vidya maybe is unable to talk. That's why she's putting in the chat box. Okay. Okay. So going forward. Okay. This is I just put this for completeness, because we talked about if you remember rolling circle replication. Here it is shown more nicely. This is this double-stranded DNA. Then you have a little cut. Then it unwinds and then releases it. And then when it is released, then after that is released, you get this strand being made kind of continuously, 5' to 3' direction. And then as this strand is released here, you get this Okazaki fragments coming in the other direction. And then they are sealed to give you pieces of DNA which are the same length as the original. And I can simply recircularize them and get more and more copies of this double-stranded DNA. And just to complete the discussion, this is the D-loop replication which I showed you, where the two strands separate. Replication begins on reading this one strand as a template, while the other strand is still not being read. Then it reaches one point, and then the replication starts in the other direction. Okay. So going to more complex systems, that is you and me. Okay. So how does replication occur in higher order systems? Okay. And the difference, as I said, between prokaryotes and eukaryotes is in prokaryotes, the DNA is present in the cytosol itself. Okay. While in eukaryotes, the DNA is present inside the nucleus. So this replication of DNA has to occur inside the nucleus in case of eukaryotes, while it occurs in the cytosol in case of prokaryotes. Okay. Now, jumping ahead a little bit. Okay, because in prokaryotes, the DNA is in the cytosol. The other function of DNA, if you remember, other than the replicative function is the function where the message in the DNA is read to give you RNA, and then later that is translated to give you a protein. Now, with respect to that process, something interesting happens in case of prokaryotes. If a DNA is being converted to RNA, okay, as a part of transcription process, this will also occur in the cytosol. So as that mRNA is being made and released in the cytosol, ribosomes can add to that newly synthesized mRNA and start making the protein. And not only that, at one time, more than one ribosome can move along the mRNA, and each one can make a protein. So this is the one which has moved the farthest. So you get a bigger protein. This has moved a little bit, you get a smaller protein. This has just started to move along the mRNA, you get a smaller protein. But what is more important is that as transcription occurs, translation can also start. Okay, so that's why they say transcription and translation are coupled both in terms of location and in terms of time. But in eukaryotes, because the first part, which is formation of RNA, occurs in the nucleus, and then this RNA is also processed. If you remember, you must have heard in 11th and 12th that introns are removed, poly A tail is added, 5' cap is added. So if all these processes have to occur, and many of these occur in the nucleus, then only after that occurs and the mRNA comes into the cytosol can protein synthesis begin, which means the process of RNA making, which is transcription, and translation are actually separated in space and time. So both occur in different locations, and always transcription occurs first, followed later by translation. Okay. So anyway, that was going ahead. But what I wanted to tell you is, in one case, your DNA is present in the cytosol itself, and in the other case, the DNA is present inside the nucleus. So that is one big difference. Okay. What is the other big difference? If you can think between, say, E. coli and you, other than location of the DNA. Anyone? The other difference? And you have a little bit of clue here in this picture also. If you look carefully at how the DNA is shown in both the figures.
Type of ribosomes?
No. I'm talking in terms of replication.
Circular DNA. Sir,
Exact.
circular DNA and we have normal
Correct.
chromosome type.
In bacteria, most of the times DNA is circular. While especially us in humans, remember we have 23 pairs of chromosomes, but each chromosome that we have is actually linear. So it is not circular. And that is kind of shown here. If you look very carefully, see this DNA, double-stranded DNA, it is going like that. It is not joined to each other, which means what I'm showing here is actually linear. And I've shown only one of the chromosomes. In fact, we have 46 like this in almost all our cells, except some cells which are haploid, which contain 23. Okay. So the other big difference is that eukaryotic chromosomes, or I should say human chromosomes, are more in number. Okay. We don't have a single chromosome. We have 46. And not only that, they are linear chromosomes. Okay. And that is where you have to remember the two differences that might end up impacting how replication occurs in how us or eukaryotes relative to how it occurs in eukaryotes. Okay. So now before we go into differences, let us look at the similarities. If you compare our replication with what happens, say, in E. coli or any bacteria, the one thing is we both require an enzyme called DNA polymerase. Okay. However, the actual structure of DNA polymerase in terms of sequence of amino acids and the overall size is slightly different when it comes to E. coli versus what we call as eukaryotic systems. but essentially the enzyme is still a DNA polymerase. The second is both E. coli and us, the polymerase which does the polymerization can only add in the 5' to 3' direction. Not only that, this polymerase in both E. coli and us also has what we call as proofreading capacity. Okay, so it goes back in the other direction in case it makes a mistake. Both the polymerases require all the deoxynucleotides to be present, and they also work better when there is magnesium. Okay. In addition to that, both the polymerases require a template and a primer. Okay. So, in that regard, conceptually, our polymerase is actually no different than the E. coli polymerase in terms of all the main properties that E. coli polymerase has. The only difference, as I said, is because our amino acid sequences are slightly different, our active sites are slightly different, but they are still similar because they still accommodate, in a sense, a double-stranded piece of DNA which is separated to make it single, and then you add a primer and stuff like that. Okay, so in that regard, we share fair similarity to the way our enzyme works and the way the E. coli enzyme works. Now, what are the differences? Okay. Now, this is some of the differences. So, differences in some case are with respect to the process, and in some case with respect to the nature of the DNA. Okay. So, the first thing is our replication is much slower. Okay. Our polymerases, okay, tend to not work at that high speed that you see in case of bacteria. So, they almost say that we are many times 10 times slower than them. Okay. We add only about 100 nucleotides per second. Okay. The other thing that you find is to make up for this slowness of speed, what you find is that in many of the eukaryotic chromosomes which are linear, there is more than one origin of replication. Okay, so Vindhya, so here is what the answer you gave, which is probably true for eukaryotic systems, where replication can begin at multiple sites along the linear chromosome, allowing for overall replication to be fairly fast, even though a given polymerase is slow. The third thing that you find is that the DNA which is present in eukaryotic systems is associated with histones. Okay, so it is packaged to an even greater extent than what you would see in case of bacterial systems, where you do not have these histones. Okay, and therefore this untangling from the histone is also part of the process which occurs or is needed when replication begins. Okay. Overall, what you find is eukaryotic polymerases are slightly bigger, appear to be a little more evolved, and their interactions with DNA are a little more complex. Okay, so the way they bind to the DNA, which is a template, and then they add a nucleotide to make new DNA. Overall, it appears to be a little bulkier and a more specialized enzyme, and maybe because of that, it is slower. Okay, and the last thing is our chromosome is linear, and therefore it requires special processing of the ends. We'll come to that a little later. Okay, although many of you are probably familiar with the fact that this linear chromosomal DNA creates a big issue when replication occurs, especially at the ends of the chromosomes. Okay. So again, going back, one, we already talked about this. We tend to have multiple chromosomes. They tend to be linear. We also have, in addition to our chromosome, mitochondrial DNA, which actually is replicated almost very similar to what happens in bacteria. The other thing is you have to realize bacteria ka entire genome is only about 4 to 5 million base pairs. while ours is, if you take out all the chromosomes, close to 3 billion base pairs. Okay, so the number of bases that we need to add is more. But the good thing is that at one time, I can have all the 46 chromosomes all replicating at tandem, and each one replicating with multiple origins of replication. So even though the speed of my polymerase is a little less, I'm able to accomplish DNA replication in reasonable time. Okay. In fact, they have calculated that if we did replication like bacteria does, and presume we had only one big, 3 billion base pair genome, that it would take years to replicate one DNA. Okay, it will be very, 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 very slow. Okay, or uh, probably not possible at all to even divide a cell. Okay. Now, look at our polymerases. Okay, if you remember, the polymerases in E. coli were called polymerase 1, 2, and 3. Okay, eukaryotic polymerases tend to be numbered as alpha, beta, gamma, delta, epsilon, and stuff like that. Okay, and here is some information, especially in terms of size. Okay, so look at this: alpha, delta, and epsilon are in the nucleus, which means they are involved in replication of our main genome. Gamma is a mitochondrial enzyme, which means that is probably involved in the replication of mitochondrial circular DNA. Okay, but this slide is slightly better. Hopefully, you can see this. What is interesting is to realize is that the DNA polymerase, which is the equivalent probably of polymerase 3 in bacteria, as far as eukaryotic systems are concerned, is this polymerase which is called as delta polymerase, okay, which has very high processivity and very high fidelity. Okay. In addition to this, there is a epsilon also, which has got the same properties, high fidelity and high processivity, but it does most of its jobs in terms of repair. Okay, so the major replicative enzyme is polymerase, which is called as delta polymerase. Okay, and similarly, this alpha does a little bit of that activity of what is called as proofreading. And look at this, it also acts as a primase. It makes a primer. Okay, so our primase enzyme is actually a variant of a DNA polymerase, which is called as alpha polymerase. Okay. So other than that, we are fairly similar. Now, this is what I wanted to show. Okay. So if this is our linear piece of DNA, what is actually observed is that when this DNA has to be replicated, and because our polymerases are generally slower, you might find that when replication actually begins, it might start here, and you have this one origin of replication and the two forks moving in either direction. But as this is happening, replication might also begin at another origin. And from there also, the forks will start moving in either direction. And when this fellow and this fellow meet, they will merge, and that will finish the replication of this strand. Okay. In fact, it can occur in even more complex fashion. So here is your parental DNA. It separates, and here you have multiple origins of replication. So each one is like a bubble or an eye. Okay, and each one is moving in either direction. And as this goes along, you'll find now the DNA will look somewhat like this. The eyes are all bigger, and then as the replication forks merge, you'll find that the DNA will get replicated. Okay, so in comparison to what we saw earlier, which was circular DNA, where there was only one origin of replication, then the forks moved in either direction, and the DNA was initially replicated to give you what is called a catenate, which got separated to give you two pieces of double-stranded DNA, where if you look, this outer fellow here is the original shown in red. That is this one. And that was replicated inside with a new strand. And its partner here, which is shown here as orange, is shown on the inside here. And the newly replicated strand is shown in orange is outside. So these are the two parent strands. Here the outer red one and the inner red one, and these are the replicated strands. Okay. Now similarly, related to this is the linear DNA. Again, multiple origins of replication, each then progressing in either direction to finally give you the two strands, where this is one parent, this is one parent, this is the newly synthesized, and this is the newly synthesized. Okay. Now, what you have to realize is that when these replication forks move in either direction and they meet here, one of these strands, which is made in a leading fashion, will merge to this other fork in which it is a lagging strand. And here, the one which is the leading strand will merge at this fork with the other fork, what is called as a lagging strand. Because remember, in both the forks, the direction of replication is still 5' to 3'. In one case continuous, in the other case what is called as discontinuous. Okay. Now, see what happens in a linear chromosome. Okay. So, here is the one bubble. For the moment, just presume that this piece of linear DNA is replicated only with one origin of replication. Okay, but what I say will apply even if there were multiple origins of replication. Okay, now remember that from this replication fork, okay, now look at the orientation of the top strand. It is 5' in this direction to 3' in this direction. So the complementary strand has to be made in the opposite direction. Now from this fork here, when the forks move here, this can be made in a continuous fashion, but in this fork, which is moving in this direction, it is made in a discontinuous fashion. But if you remember what I told you about Okazaki fragments, when one Okazaki fragment encounters another Okazaki fragment, because of the ability of this polymerase 1, or in our case, the DNA polymerase alpha, this RNA primer is removed, and that RNA primer is replaced by DNA because of the 5', 3' exonuclease activity, and then it is sealed by ligase. So what will happen is these two Okazaki fragments will become one. Not only that, this Okazaki fragment is going to encounter the primer of the leading strand which started from here. And what that will do is remove this RNA, add DNA, and seal so that at the end will go all the way and complete the replication. Okay. Now that is not the issue. The issue is on this side. Now imagine the last primer that was made, from which you have a Okazaki fragment, and that Okazaki fragment moves in this direction. Now that can go ahead and touch the previous primer, the Okazaki fragment, and do all the replacement and sealing, and have a continuous piece of DNA. But at this end, what I'll find is a little piece of RNA. Now, I do not want this RNA, right? Because my genetic material is actually DNA. And the other thing you have to remember is RNA is much more unstable than DNA. So after a while, what happens is the RNA simply, because of its degradative capacity, gets automatically lost from this end. And since there is no polymerase which can add without a primer, this end will never get replicated. It'll stay as a little gap. And the same thing is going to happen on the other strand with respect to this direction, where this last primer which was added here will have a piece of RNA which can never get removed because there is no polymerase coming from here because there is no DNA left. So over a while time, that RNA will also get lost. So in the end, I'll have gaps in the newly synthesized DNA, which means the newly made DNA will have little gaps at the 5' end of one of the newly synthesized strands and the 5' end of the other newly synthesized strand. That means I'll get shorter DNA than what I had with respect to the parent. And this problem is referred to in DNA replication parlance as the end replication paradox of linear chromosomes. Because of this unique requirement of the polymerase to always need a RNA primer to begin its addition or making of the DNA polymer, when you come to the end, you'll have no way of replacing this last primer ka RNA piece that you have added. Okay, because there is no more template left to make another piece and then fill it. Okay, and then now what to do? So my new DNA is going to be shorter. Okay, and this is also shown here. I have this fork here. This went like that. This kept being made as Okazaki fragment. But what happens to the last one? Okay, no DNA for another priming event. Okay, either high in na. So once this primer is removed, I cannot actually make new DNA come and remove things. Okay, here there is no problem. I can keep removing because there's always a piece of DNA I'm dashing into it. So I'm just removing the RNA, replacing the DNA, and sealing it. And here I have no issue because I have gone all the way to the end. Okay.
So
Yeah.
So you said that the RNA primer gets removed and then it is replaced by the DNA fragment.
Correct. But
So
who
how it
puts?
is?
Yeah. Go ahead.
So how it is like shortened we get at the end?
See, now here, when I remove the RNA primer, correct? Look at this point here. Can you see my arrow?
Sir, actually your this pointer is not visible.
Take a second. Let me see. Okay. So here are the RNA pieces of the previous Okazaki fragment. Okay. Now, when my polymerase is coming in this direction and it hits this RNA primer, my DNA polymerase, which is polymerase 3 or polymerase delta, will stop. Then the other polymerase, which is polymerase 1 or alpha, will come, and that has 5' 3' exonuclease activity. So what does it is removes the RNA. Now after it removes the RNA, it replaces it by DNA. But in order to replace it by DNA, it has got a 3' OH here, no? And to that 3' OH, it starts adding DNA. Because now this piece of DNA actually acts as a primer for the polymerase. It fills it with DNA, and this nick which is there is sealed by DNA ligase. Similarly here, I have no trouble filling this with DNA because I have a 3' OH to add. Now see what happens to the last primer. This piece of RNA, when it is removed, if I want to replace it with DNA, my polymerase has this unique problem that it can only add to a 3' OH end. It cannot start from nothing. So what happens is, since I have got nothing here, I'm unable to fill this gap with new DNA because I have no 3' OH to use to add the new DNA. While here I always had, even when I removed that RNA, this strand ka 3' OH was there, no? To which I added new nucleotides. So that acted as a primer. Okay. Is that clear? Am I making sense? Okay.
Yes, sir.
Now, so in all these cases, to fill the gap was no issue because there was a 3' OH which was available to which the polymerase could add things. But when I came to the end, when I removed this primer and I want to now fill it with DNA, I got no 3' OH to add because this is the 5' end. And my DNA never grows in 5' to 3' direction. Okay? And that is why I end up getting a piece of DNA which is shorter because that primer has not been filled because there is nothing to polymerize the DNA on. And my DNA polymerase is this unique fellow. It needs not only a template, but it also needs a 3' OH. I have template here, but I don't have a 3' OH to add to. Okay, and that's why I am unable to replicate the end. Okay. Now, how do we solve this problem? Okay, normally, okay, and this is very, very interesting. Many of the cells in our body do not solve this problem. Okay, so when they replicate, no, the new daughter DNA is slightly shorter than the parent. Okay, and you might think that that could be an issue. Okay, yeah, it could be an issue. Okay, but what has happened over time, okay, as part of evolution, is that bulk of the message which is present in DNA resides here. Okay, so even if when DNA gets replicated, it becomes shorter by maybe about 20 to 25 bases, which is probably the length of the primer. Before the replication events can occur many times, and I end up getting a relatively shorter piece of DNA relative to my original parent and start creating problems because now my function is being affected. That number of replications never occurs in a human. Okay, so when cells are produced which do not have to replicate too many times in a lifetime, and they have to replicate only a few times in their lifetime, even though every time they replicate, the DNA which is produced is slightly shorter than before, the overall functionality of DNA is not affected because most of the genes are present in this region and not at the ends. However, we also have in our body lot of cells which need to replicate many times. Okay, for example, the cells of our bone marrow. For example, the cells which line our intestine. For example, the cells which make up our hair. For example, the cells which are used to produce sperm and ova. In that, the number of replication that occurs in a lifetime is significant. And therefore, if something is not done to take care of this problem, what you'll find is as you get older and older, those cells which are replicating will end up producing DNA which is shorter and shorter because they've undergone many replications. And then the DNA which is being produced is so short relative to the original DNA that it has got compromised function. So these type of cells which need to replicate more often than the other cells have over the years evolved to take care of this end replication problem. Okay, and the way they do that is by invoking an enzyme called telomerase. Okay, and telomerase is an enzyme which helps take care of the problems associated with what we call as telomeres. Okay, and what are telomeres? Telomeres are those sequences which are present at the end of these chromosomes. Typically, what we call as the two ends of the chromosomes. The sequences which are present there are very, very specialized sequences, and the sequences are referred to as telomeres. And the enzymes which work at this region to take care of the issue of these telomeres are referred to as telomerases. And telomerases are very interesting enzymes. They are what we call as reverse transcriptases. Has anybody heard this term, reverse transcriptase?
Yes, sir.
Sir, in
Related to
HIV.
the
RNA to
HIV.
DNA.
They take RNA and from RNA they make DNA. Okay. For example, as shown here, if I have single-stranded RNA and if I'm able to use this single-stranded RNA as a template in order to make a complementary strand of DNA, then that enzyme is referred to as a RNA directed DNA polymerase, or sometimes it is called as RNA dependent DNA polymerase, which means it is able to make a DNA polymer, but it does so by reading RNA as a template strand. So when it is finished, what you end up getting is what you call as a DNA RNA hybrid. Okay, is that clear? For the moment, I'm not going to tell you what this part is. Okay, we'll learn that in rDNA technology also a little later. But hopefully you recognize this enzyme. See this enzyme RNase H. Remember what it does? Anyone? RNase H from our rDNA class.
So it removes the RNAs from it. Like if there is any single-stranded RNA which is present in the DNA, it will be removing that.
So see what it will do to this? It will remove all this red, which is single-stranded. So what you'll end up getting when you treat this hybrid with RNase H is a DNA, but this DNA is going to be exactly complementary to this RNA. Okay? And let us say, then I take this DNA and convert it into double-stranded DNA using a DNA-directed RNA polymerase. By giving it an appropriate primer at this end, I'll be able to get double-stranded DNA from single-stranded RNA. Okay, but that is besides the point. What is important is this reaction here, where I use RNA as a template and make complementary DNA shown here in blue. Okay, now telomerase, the enzyme that I'm talking about, uh, is an enzyme which actually is a reverse transcriptase. Okay, so see what it does. Okay, now telomerase is not a simple protein. It is what is referred to as a ribonucleoprotein. What do you mean when you say ribonucleoprotein? Basic biochemistry, ribonucleoprotein.
Ribose sugar and with a
Hmm.
nucleotide or nucleotide
Okay.
base.
Okay. So ribonucleoprotein is a protein which is actually a mixture of RNA and proteins. Remember, we have ribosomes. Ribosomes.
Yes,
When we
sir.
talked about, we said it's a complex of proteins and RNA, and we said 16S RNA, 23S RNA, and stuff like that. Okay. Similarly, this telomerase is also a entity which is a combination of a protein part and a RNA part. Okay. Now the interesting part is this. Here is that telomerase, and what telomerase has is this green thing which is shown and that is a protein part of it, and inside that is a piece of RNA. And if you look at this, you see the sequence CAAUCCC. So the minute you see U, that tells you that it is not DNA but it is RNA, because RNA is made up of A, U, G, and C. But what is very interesting is that this enzyme has this little piece of RNA which is exactly complementary to the ends that you see in the chromosomes. Remember, I told you the ends of the chromosomes are called as telomeres, and the reason they are called telomeres is because they tend to have a lot of repeating sequences which are exactly the same as the end. And this relates to what I told you earlier. Remember, I told you that much of the gene information is in the middle of the DNA, while towards the end, you don't have actually a lot of information. But what you have at the end is these repeats. Okay, in this case, look at the repeats that I have. The repeats that I have is C, TTA, GGG, TTA, GGG, TTA, GGG, and so on and so forth. So these sequences ends up getting repeated at this end. And similarly, you'll find at the other end also the same sequences are repeated. Okay, but on this other strand. Now, this is the part, remember, which is not replicated. Now, see what this telomerase does. The sequence of RNA that it has is exactly complementary to the sequence. So, what it does is it binds to this end, and because of this complementarity, see what happens of the way this RNA associates with this end. There is a C, A, U, C, C, and so on and so forth. So there is a similarity. So they now form a RNA DNA hybrid. Okay. Now, when this happens, you realize now I've got a 3' end here, right? And this enzyme, which is a reverse transcriptase, reads this RNA, which itself it has got, and it uses this DNA, which is bound here, and extends that DNA, okay, by adding complementary bases. And not only that, it does that many times. So once it adds here, it'll move a little bit. Again, this RNA will bind to this complementary sequence. Again, new DNA will be added. So what it essentially does is it takes this 3' end of the original parent and makes it bigger. Okay. Now see what happens. Once this strand is made fairly big, then my primase enzyme can actually come, add a primer somewhere here, and then my polymerase can come and add my DNA all the way up to this point. Okay. Now, by doing this, what has happened is essentially my original DNA has been completely replicated. But in my enthusiasm, what I must have done also is get a new strand of DNA which is actually slightly longer than the original. Okay, and that actually happens. Okay, so in cells which have this activity, which are called as telomerase activity, not only is the issue of getting shortened strands solved, in fact, when telomerase works, many times what you end up getting is both the parent and the newly made strand are slightly longer than what you expect them to be. And therefore, no information from the end is lost at all. Okay? And therefore, even if multiple replications occur, the DNA never becomes short enough and it never loses the functionality. And if it does become big, remember, it has not added new information. All it has done is probably added more of these repeated sequences, which do not have any consequence with respect to the functionality of DNA. In fact, something interesting is observed. When a normal cell, which doesn't replicate that often, actually becomes cancerous, what is observed is that the cell somehow acquires this telomerase activity, which means that when the cell replicates more and more times, it makes sure that its DNA never gets shorter in order to compromise the function of the cancerous cell. So, lot of times, treatment of cancer is accomplished by taking what are called as telomerase inhibitors, which are actually now produced by these cancer cells in order to allow the cancer cells to keep on replicating without shortening the end of DNA. Okay? Does that make sense, everyone?
Sir?
Yeah.
Yes, sir.
Sir, as it is forming a hybrid inside the telomerase, so RNA polymerase will be working there, right? To break that so that primer can add.
Yeah, see what is going to happen here is this is the RNA which is bought by the telomerase. Okay? This is the new DNA which is actually made by telomerase. Okay. Now once it adds a bunch of them and makes this strand slightly bigger, then telomerase actually leaves. It just goes away. Okay. And now our regular system, remember the primase or DNA polymerase alpha comes somewhere here and makes a little primer. And after it makes a little bit of primer, it then recruits a DNA polymerase, or it itself has a capacity to do it. It'll add a new DNA here. Okay, and it'll add the new DNA, it'll go all the way, come here to this point, and then it reaches here, and then DNA ligase seals it. Now remember, our original DNA was only up to this point. So I have actually made my intact DNA now. Now, when I come to this end, remember there is a RNA primer here. That primer will get removed, but that will never get replaced. My end will look funny, but the important part is it is now bigger than my original. So in a sense, I prevented damage to my original DNA. I've kept that intact, but still what I have made as double-stranded DNA will have one strand which is longer and one strand which is shorter, because this primer will never be removed. Okay? Is that clear? Does that make sense?
And so, how long the telomerase will work? How long it will extend the DNA?
That is a little bit variable, okay? Uh, because there is no template which is going to direct when to stop, in a sense. Uh, it all depends on number of times it keeps on jumping like that, okay? And that sometimes can occur maybe about 20, 30 times or 40 times, but it is never very consistent. So you'll find that different chromosomes, when they are acted upon by telomerase, will end up getting extended to slightly different lengths, okay? Uh, but they will be slightly variable, okay? But I can't give you an exact answer, how many,
Uh,
22 or
so,
23.
sir, is
But it
this the
adds,
reason? Is this the reason that like the 23 chromosome which we have are different in sizes?
no, 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 no. Each of those chromosomes, when you are born, has got no problems with the 3' end. They are all intact. Okay, no issues there. Okay, uh, but because that is the information they carry. Okay, later on when they replicate in our body, then their sizes might end up actually being slightly less. Okay, now when I talk about replication to give you haploid chromosomes, then you have to realize that the main information part is never compromised, because the replicative cells will never have DNA which is shorter, because they have telomerase activity. So you don't have to worry of that many generations down the road, some chromosomes will disappear, because every time I pass on to the next, I might shorten it. It doesn't happen, because the reproductive cells, they have telomerase activity, so their shortening does not occur. Okay,
Yes, sir.
is that clear? Yeah.
Yes, sir.
So our
Hello, sir.
chromosomes, yeah.
So
Hello.
sorry to disturb, but I had a doubt.
Mm-hmm.
Sir, I had read somewhere that while aging, the telomerase activity and the telomere shorten, and that's why we age. Actually, that was something that I read. So, is it actually
Yeah.
related to the telomeres that gets shortened every time, or the activity gets, uh,
Yeah. See, I told you before, right, that many cells in our body do not have telomerase activity. Okay, uh, and these are cells which typically, as we go through our life, are not cells which replicate very frequently. Okay, the cells in our body which replicate frequently and which tend to have decent telomerase activity are actually cells which are of the bone marrow, cells of the reproductive tract, etc. But many other organs which also undergo replication during our lifetime. For example, our muscles grow, our heart ka size grows, our pancreas ka size grows, our diaphragm size grows, our bone ka size grows up. Those cells do replicate, but they replicate at a much lesser frequency than the replication occurs in these other cells. So their telomerase activity is a little low. So as we get older and older, okay, and as we continue to make, rejuvenate our muscles, rejuvenate and take care of some tissue damage, cells which normally do not have very high telomerase activity now get impacted, because as the number of times my cells are dividing and my telomerase is getting shorter and shorter, or my chromosome is getting shorter and shorter, then at one point important genes which are actually present at the ends of the shortened chromosome, now their function gets compromised, and therefore I start getting issues with the functions of many organs, and that reflects many times as what you see as a aging process, okay, where you find regeneration doesn't occur as fast as destruction of a tissue occurs. Okay, so a lot of people feel that telomerase activity, shortening of chromosome, is in a way related to what happens as we age, and then therefore one way to prevent aging or becoming younger is to take what are called as telomerase inhibitors. Okay, but that is still a theory. Okay, maybe it might work out, maybe it won't. I really don't know, but they say, okay, one way to treat aging and preventing this, loss of function of different organs is to maybe give telomerase inhibitors. Okay. Sorry, I should say not telomerase inhibitor, but to impart what is called as telomerase activity to these cells also, so that they do not cause damage to the ends. Okay, I said it in the reverse. Okay, so you give what is called as agents which are telomerase-like activity, so that my ends are always preserved. But that is very difficult to do. How will you deliver telomerase activity to all these cells? Okay, it's a big protein. It's a nucleoprotein. Not probably very easy to do. Okay, but yeah, that's a theory. You are right that it is related to,
Thank you, sir.
in a sense, aging.
Yes, thank you.
Okay. Any other question? Okay, I'm going to stop sharing and see what we call as a at the chat box. Okay, let me see. Okay, anything in the chat box here? Yeah, in cells which do not have telomerase activity, there is still the 5' end which says single stranded. And what does it say? And single stranded DNA are more prone to mutations. So it won't create a problem in the cell, and won't that free end create problems while coiling with histone? Okay, one part I answered, right? That because of telomerase activity, your DNA will end up getting longer. But as you correctly pointed out, the end will still be single stranded. Okay, and therefore, whenever DNA is single stranded, you might have issues that it is more prone to mutations. Okay, that's very, very correct. And will it create problems while coiling with histone protein? Okay, actually, no, in a sense, because another theory says that this end, okay, which is now slightly single stranded, there are specialized proteins which actually bind to this end on both sides. Okay, yeah, Shivam, correct. Both sides you will see the activity. But as to answer Aditi's question, at the end, where you have this slightly free ends, specialized proteins actually bind to this end, so that this mutation and this, funky structural features issues are prevented. So Aditi, you are right that single-stranded end, even after telomerase activity, can potentially cause a problem, and that is prevented by proteins binding to the ends of this telomerase and stabilizing this what is called as single-strand overhangs. And therefore now even histone interactions will not be an issue. Okay. Does that answer your question, Aditi?
Yes, sir. Thank you, sir.
Yeah, yeah. Okay. Let me see. Okay, I need to download your attendance, so that is done. Can I now stop the recording? Any other questions, anyone? Anything else in the chat? Let me see. No, I think I answered the main questions. And Shivam, your question is also answered. Telomerase activity is seen at both ends.
Yes,
Yeah.
sir. Yes, sir. And sir, can
Okay.
you tell the books that we can refer for all this biochem?
Yeah, yeah. I'll put it both for this and RDNA. I'm trying to find out which is the easiest and more, nice to read also. There are many books. So I'm trying to pick one which you might like reading, and which is covering things at the depth you need rather than being overwhelming. Okay. Many books, when you first read them, they have so much information you don't know what to read and what not to read. Okay. So let me figure out which I should suggest to you. Okay. I'll have it to you maybe by next week. Okay.
Thank you, sir.
Okay. So I'm going to stop my recording now, and